Okay, everybody, so the purpose of this video is to learn how to draw 3 kinds of cells. All living things fall under one of 3 domains. We talked about these in class. Those are archaea, or extremophiles. Then there's prokaryote, which encompasses all of the bacteria, and eukarya, which are those complex cells that make up plants, animals, and fungi. We are not going to be worried about drawing archaea. We are going to learn how to draw a type of prokarya and eukarya. For prokarya, we're going to learn how to draw E. coli as an example. Notice how I underline that. It's the proper way to write the scientific name of a species. And then for eukarya, we're going to learn how to draw 2 kinds of cells. The first kind is an exocrine gland cell. An exocrine gland cell, exo meaning outer, and crine has to do with secretion, I think. But anyway, these are things that make a whole bunch of stuff that gets secreted out of the cell. And what's called a palisade mesophyll plant cell. These are the cells in plants that do photosynthesis. So for this first video, we will learn how to draw an E. coli cell. Now, an E. coli cell is a long, thin cylinder with a whip-like tail on the end of it. It has some simple hair-like structures on the outside of it. I'm going to draw it one way, but how I draw it, the orientation of it isn't important. If you draw yours with the flagella coming off the right end and going like this, or if you were to draw yours with the flagella coming off the other end and looking like this, like both of these would be fine. But the point is that the right stuff is in the right place, but it could be facing in any direction. Now, you don't just have to draw, but you also have to label all of the different parts of the E. coli, and you have to annotate those parts with respect to what their functions are. So, let's draw an E. coli. So like I said, it's kind of like a long cylinder. It's kind of like a long cylinder like this. And there's a whole bunch of parts of it. So let's label the first part. This right here, this is the plasma membrane. We'll learn a lot more about this plasma membrane this section. But the function of the plasma membrane is to control what goes in and out of the cell. Sorry for any of the background noise going on. Everybody's at home. We've got laundry going and dinner's cooking and all that kind of stuff. Okay. That is the plasma membrane. On the outside of the plasma membrane is and there goes the washing machine, is a thicker cell wall. And I draw this nice and thick here. I'm going to give it some shading just so that you can see that it's different from the plasma membrane. Make sure that when you draw this, that it's like really clear to see that the cell membrane is on the inside of this cell wall, and the cell wall is all around the outside of the plasma membrane. All right, so this is a cell wall. It's unique in the fact that it is not the same as a plant's cell wall. You might remember that plant cells have a cell wall, but it is not made of the same stuff as a plant cell. But the purpose of this cell wall is to give the cell structure. so that it can keep whatever shape it is, and it gives the cell protection. This is really important. Maybe you've heard of penicillin, but penicillin is an antibiotic, and what it does is it prevents the cell wall from forming very strongly so that the cell gets ripped apart as it travels through your bloodstream. Okay. On the outside of this E. coli, there's lots of these little tiny hair-like structures. Each one of these singularly is called a pilus, but altogether these form what are called the pili. And in E. coli, these pili have one specific focus, and that is to help them adhere or hold on to surfaces. Different kinds of prokaryotes have different kinds of E. coli make pili for different reasons. Sometimes they make them to hold on to other bacterial cells that so they can reproduce with each other. Some of them actually use them for movement, but for E. coli, it uses them just to hold on to cell surfaces. Attached to this E. coli is also a really long tail that functions a lot like a whip. It helps it move around. This has the same name as the tail of a sperm, but it doesn't mean that our sperm are E. coli. It just means that they have the same part. This is called a flagellum, and this moves like a whip and helps the E. coli move. That's pretty much it for the outside of the E. coli. Let's go to the inside. Everybody hold on one second. I fed the cats. Good, now you all know I fed the cats. Okay. All right, let's talk about the DNA inside of an E. coli. Prokaryotes all have a single piece of DNA that is just kind of all jumbled up like that. But I want you to notice that when I drew it, as I drew it, I could trace this path and it ended where it started. That's because DNA inside of a prokaryote is circular. Not like ours, which is just a really long string. So this is the DNA. In this area that the DNA can be found in is called the nucleoid. Now we'll learn a lot more about DNA later on, but for the time being, you just need to be able to label these things and not list any kind of function at all. There is a liquid that everything on the inside of the cell floats in. That liquid is called cytoplasm. A lot of chemical reactions happen here. I didn't give myself a whole lot of room. I might need to say some things right now, and you'll have to write them down. But it contains all kinds of dissolved substances. A lot of chemical reactions happen here, and so on and so forth. Okay, I'll give myself some more room to write some things. There's a lot of little tiny structures inside of an E. coli that you've probably heard of before. We can draw a whole bunch of them, and they're just, for our drawing purposes, they're just little dots. But these are called ribosomes. They're not surrounded by a membrane, so we're not breaking any rules here. But each of these dots is a ribosome, and they're just a little set of RNA, but their purpose is to assemble or put together proteins. And we talked about proteins already. They do all of the work inside of the cell. And so these ribosomes are just floating all over the cell, just constantly building the proteins that this prokaryote, this E. coli, needs to survive. And then finally, our last one is we're going to draw a couple of circles in our cell. And these little circles are actually circular pieces of DNA that are called plasmids. And plasmids are little circular pieces of DNA. And these pieces of DNA are really interesting. And humans have learned to use these pieces of DNA to our benefit. But these little circular pieces of DNA normally just contain enough information for one particular characteristic. We call that characteristic a gene. And a lot of times that characteristic is like a resistance to some kind of antibiotic or something like that. These plasmids can often get traded in between E. coli or different bacteria, so that antibiotic resistance can actually be passed on. Okay, that's it for drawing prokaryotes. The next video will be about how to draw an exocrine gland cell, which is a eukaryotic cell.